Right, so good morning, everyone, and welcome to the Center for Theoretical and Computational Physics weekly seminar. And today it is a pleasure to welcome Daphne Klotzer from University of North Carolina, Chapel Hill. And well, a few words of introduction. Daphne did her undergraduate studies at University of Warwick in the UK. This was followed by a PhD at the University of Nottingham, also in the UK. And then she held a number of postdoctoral appointments. And I hope I'm going to get this in the right order. Was it first Bath, then Michigan, then Harvard, then Cambridge? Is this more or less right? Okay, and then she got her present position at the University of North Carolina. And today she's kindly agreed to tell us about her work, somewhat intriguingly titled A Touch of Non-Linearity in Living Matter. Before I hand over to Daphne for the truly exciting stuff, just a few announcements from me. First, this seminar is being recorded, so by staying on, you're giving your consent to your voice and image being recorded and stored. And second, in the interests of internet stability, can I ask everybody except the speaker, of course, to mute their microphones and turn off their cameras during the seminar. If you wish to ask a question, then of course you're free to unmute yourself and turn on your camera if you so desire. Okay, so now I'll shut up and it's over to Daphne. Thank you.
Thank you. Thank you so much for the invite. It's really exciting to be with you all, even virtually. I wish it was in person. I'm, as I was telling Paula, I'm from Greece. Yeah, it's just really nice to be in Europe, even virtually. So I haven't been able to travel in a long time. So yeah, so I'll get started. Please ask me questions. If you have questions during the talk, I'm really happy to answer. And yeah, I think I'm a physicist as well, as Paolo said, but I am working in something that is a bit, has a lot of touches upon other disciplines as well. So if you're not familiar with things like, please, I'll do my best to explain things clearly, but do interrupt me and ask me questions, because I really like it when people can follow the talk. So especially if you're a student or a postdoc and you're shy, just don't be, basically. So anyway, so yeah, so my name is Daphne and I'm at the University of North Carolina in Chapel Hill. I'm in a department called Applied Physical Sciences. So this is an interdisciplinary department. We have a lot of different people with many different backgrounds. So we have, I think, well, we have a couple of physicists, but we also have material scientists, engineers, mathematicians. So it's a very interesting environment to work in. And today what I'll be talking about is mesoscale swimmers and active matter. And I'll just explain all of this, but I want to take a moment, because you're all physicists. I don't usually do this, but I should. I want to advertise this series that I'm organizing together with a couple more people in, so we are part, if you are part of the American Physical Society, we in particular have this group that's called GSMP, that's a group in statistical and nonlinear physics. So if you identify with this, we are organizing these series that's called KLOCW, and they're online. So you can join our mailing list, and we have Lenka giving a talk on May 20th. All our talks are also recorded and uploaded on this website. And also I want to encourage you to become members if you want of the group and APS. Anyway, so with this advertising, I will move on to just show my group. We haven't had the chance to have a photo taken in the last year and a half. And so and so anyway, this is my group with pasted images of my newer students. And this is a group website if you would like to check more of the research that we do. And here's some acknowledgements of the people that have worked on the projects I'll be talking about. So people in my group and in Chapel Hill over here, as well as people elsewhere, Laura Miller, Amnid, and applied math collaborators, and also some people in Nottingham that my advisors and collaborators from my PhD. Okay, so I would like to get started talking about I always start my talks with this slide where I'm showing different images of what we call active matter systems. And I will go ahead and define what I mean by this, but just to point out some of these, what these systems are. So here's a school of fish. These are ants. These are little robots. We have penguins, bacteria, humans, intracellular things that are happening. And these are all systems that we call active matter. And I will go through some of the characteristics that sort of in the community we have sort of agreed that this is what active matter means. So with the term active matter, we're talking about a collection of motile self-propelled particles. Okay, so so this is in contrast to things like a colloid, for example, that is undergoing Brownian motion. So these are, when we talk about active matter, we have things that are self-propelled, right? So they're not just doing Brownian motion. So energy for this propulsion is consumed locally. So this is a big difference between active and non-active systems. So each particle or each organism consumes its fuel on the local level, right? So it creates its own, it uses up energy locally. And another thing that's really interesting is that when we look at the collective behavior of these systems, what we realize is that the interactions between the particles or the organisms are often quite simple, and they're local. And by local, I mean that often what we find in these systems is that the interactions are only between immediate neighbors or pretty close neighbors. And they're not necessarily very long range. Yet what we see is when they do interact is that we see very complex global behavior. So even though the interactions are quite local, the behavior that emerges is complex and is quite large scale. So one example, typical example, is a flock of birds, right? So the interactions are, birds don't really know what's happening at the other side of the flock, but the whole structure is quite large in size. And another thing we can say about active matter systems, and this is what I'm also trying to demonstrate with these images here, is that we see this kind of behavior across length scales. So from very small scales to like quite large scales, and also environments. And by environments, and I'll be talking more about this. So for example, some things happen, in air, some things happen in water. In some cases there's friction, there isn't. So the environment can change, and yet a lot of, we see a lot of similar behavior. In particular, what I'll be focusing on is the role of the fluid environment. And what I will try to demonstrate is that there's a particular kind of region of phase space where the fluid dynamics can be very interesting. It's highly, it's not explored, especially when it comes to active matter. And but it can induce very interesting collective behavior, very interesting structures. Also, another thing to add is, especially when you think about this from a physics point of view, is that these are non-equilibrium systems. Right? So in my group, we study in general active matter in different environments. And this is another project that we're working on, that is active matter mixtures. This could be of interest to some of you. These are, this is a simulation of fast and slow self-propelled colloids. So there are thousands of particles in here, and they mix and they interact. And there are many interesting questions that arise when you think about mixtures. So self-propelled colloids, so you can imagine we have these sort of self-propelled nanoparticle, colloidal particle. So most of the work that has been done in this active matter community, and a lot of it has been theory and simulations, is to show interesting collective behavior, interesting phase transitions, non-equilibrium phase transitions, but it's only been done for monodisperse systems in general. And what we are really interested in is looking at mixtures of fast and slow particles. And if you think about it from a physics point of view, there are many interesting fundamental thermodynamic questions that arise. So imagine you have a substance that you have it hot and cold, and you bring them into contact, and the system equilibrates into one temperature. When you have active systems where you have fast and slow, when you mix them, they don't, their velocities are not going to equilibrate, and the question is, what does equilibrate? What happens in these systems? Can we understand the sort of the thermodynamics, and what does it mean in terms of non-equilibrium physics? So there are some interesting questions there, and we have a paper published. We have one submitted recently that's on these mixtures. And yeah, this is one of the projects that I'm working on that I won't be talking about today. And we also have things that we work on that's more related to biology. This is a liquid-liquid phase separation in the cell. These are little droplets that form that look like oil and water, but they happen in the cell where there's a lot of stuff. And these can also be quite thought of as out of equilibrium. And there are a lot of active processes in the cell because, of course, cell is not at equilibrium. So there are a lot of interesting questions there as well, but I will leave this for another time. So today I'll be talking about active matter and fluids, and in particular, what we call the intermediate Reynolds range. And so what I would like to start with is, so in biological fluid dynamics, which I don't expect people to be familiar with this. So I'm just going to go ahead and explain everything, but please do interrupt me if there are questions. So in this field of looking at motility, swimmers and flyers and things, little organisms that move in fluids, there's this plot that is this way to plot things, which I'm showing here, which is basically the velocity, the speed of the organism, how fast it swims or flies, as a function of the Reynolds number. And the Reynolds number is defined here. So the Reynolds number, when it comes to motility, can be defined as the speed of the organism times its size, the size of the organism, divided by the viscosity of the surrounding fluid. And so this is a dimensionless ratio. And so when the Reynolds number is very small, what we have is we basically have viscosity dominates the system, okay? And this is what we call the Stokes regime. So when viscosity dominates the system, so this is a region in phase space where we have very small things usually, so the length scales are also quite small. So we have microorganisms and bacteria and cells and nanoparticles and colloids and things like that. So this is when we have things like that, we always operate here. On the other limit, we have what we call the high Reynolds number limit. And here, inertia dominates over viscosity. And this is a regime where we are more familiar with as humans. We operate here. So everything like us, birds, whales, ships, airplanes, all of these things operate at the high Reynolds number limit. And here inertia dominates. And what I mean by inertia is when you swim, you push fluid behind you, and even when you stop pushing, you're still moving forward, right? So this is the inertial effect, in this context of swimming and motility. This is an example. But what I would like you to leave with today, if there's one thing to remember, is that there are also these intermediate Reynolds numbers. And here we have the influence of both components that matter. We have viscosity that matters and also inertia that matters. So we can't make any assumptions. So in the two other limits, we have the Navier-Stokes equations, but here we can neglect inertia and we can solve Stokes equations. And over here, we can ignore viscosity. But in this intermediate regime, in this intermediate range, we cannot do that. So we have to include all of the terms in Navier-Stokes, and this is a harder sort of numerical problem, and also very hard to solve analytically, and in very specific sort of cases can it be solved analytically. But what I would like to yeah, so we have these, so in this region here where we have the intermediate Reynolds numbers, this is not a region in space that is empty of organisms or processes or machines that one would like to make. So this is roughly a, just so that you have some intuition, this is roughly a size range, like 0.5 millimeter to 50 centimeters. And its organisms are like ciliates, plankton, insects, small animals, small fish, but also drones, robots, and also processes that operate at these, there are lots of processes that operate at intermediate Reynolds numbers as well. The things that I would, so what I won't be talking at all about high Reynolds numbers. So in general, I focus more on this intermediate Reynolds range and also this Stokes regime over here, close to this boundary in particular. So as you can maybe notice here, like there are these dashed lines on this side. So where the transition between the Stokes regime and the intermediate Reynolds regime happens is really not clear for in general and in particular, like in motility. So if you open a fluid dynamics book, what you will see is that we know where the transition is, but this is for, you know, flow past the sphere, which is usually sort of the textbook example. But when you have complicated geometries, complicated, you know, flapping of weird appendages, like you can see here, where this transition would happen is not clear at all in terms of where that happens. And as a sort of, again, like going back into intuition, if you're not familiar with fluid dynamics, what is happening here in terms of the physics is that we go from a regime where inertia plays no role at all because the particle or the organism is very small, into something where inertia starts to play a role. And the way this comes in terms of the math is that you have more, as you increase your Reynolds number, you get more and more nonlinear, okay? So you add more and more nonlinearities into the system. And so one big question that I'll be talking about is what happens at the boundary. And like I said, the boundaries are not clear where they are, but they can be very important both for biology and for applications, other applications that we may be interested in. And I'll just show you an example. So this is a paper in ecology. And what they are talking about here is, so the reason why I'm showing this is to demonstrate why the Reynolds number boundaries, these boundaries I was talking about, is important. And in particular, what it means, say, for a living organism to live near this boundary. So this paper, this paper was published a few years ago, and it's by an ecology group. And what they're looking at is this problem, which is that 99% of larval fish apparently die within their first few days of life. And for a long time, it was not known why. What they claim in this paper is that the reason why this happens is because of what they call hydrodynamic starvation, and I will show you what this is. But basically, it relates to the Reynolds number and how, and in a way, like which side of the boundary you're on, the boundary that I was just showing. So this is a larval fish, and I'll play this video, and this is all food, and you'll see what happens. So the fish opens its mouth and it sucks the food in, but as it closes its mouth, it goes out again. So this thing happens back and forth, and so the fish cannot eat. And the issue with this is that there's, you know, in Stokes flow, the system is linear, completely linear. There's time reversibility. So unless you break that unless you break that reversibility, if you have reciprocal motion, you don't break that reversibility, that you can't get a net sort of flow in this case. So the food goes in and out, but you could imagine trying to swim doing this. So you wouldn't, you would just go back and forth if you're on this side of the boundary. But if you're on this side of the boundary and you have enough inertia, you could do it which is presumably what, one of the things that happens when the fish is larger. So I would like to go through a few points of why this regime is interesting for active matter in particular. So I was showing you for one fish, and this plot here is showing single organisms, but I will build on why this is interesting for active matter, which is the goal in the end. So one question is what happens at the boundary, and this is what sort of I was showing you about the little fish trying to eat, but it can also mean things like going from immotile, so something that cannot swim, to something that can swim. So you could imagine having a lot of particles doing this at the same time. You have very different behavior. Another question is what happens for many swimmers? And by this, saying here on either side of the boundary, what I mean by this is that when we look at, in this intermediate Reynolds range, something very interesting happens, which is that, as I said, as you increase the Reynolds number here, you can just think about it that we're increasing the inertia of the system, and the inertia and therefore the non-linearities of the system, and it's very interesting because you get different behavior at different intermediate Reynolds numbers. So because the intermediate regime is not a limiting case, it's basically everything that where we cannot make the other two assumptions. So we can't say that viscosity dominates or that inertia dominates. So everything else is sort of in this bucket of intermediate Reynolds range. And but there's no unifying or universal behavior other than you sort of have to look case by case. And that means that, first of all, there's a lot of richness in behavior. And it means that, what you get at Reynolds, 1 is not what you get at 10, is not necessarily what you get at 100, right? So within this range, you can get very different behavior. And some of this I will demonstrate in this talk. Another reason why it's really interesting to look at this regime for active matter is because it's actually going on the other side of the boundary. So over here, so let's say here we have microscopic swimmers, so smaller swimmers on the individual scale. But if you think of swarms, so if you think about this active matter kinds of systems, when we calculate the Reynolds number to make this plot or to decide which side of the boundary we're in, we usually use the length scale of a single swimmer. But if now we have, one bacterium, it's pretty small, but if you have 1 million bacteria that are swarming, that are all swimming together, the question arises whether this length scale is more relevant and what would that mean, and whether it's something that can push you on the other side of the boundary. And of course, it becomes more complicated, but these are some of the questions that I'm interested in. I think it's really interesting to think about active inertial suspensions as materials. So if, like I said, being close to this boundary gives us more control. If we can control the system, we can cross this boundary in a way that, through a specific control in the way that we want to do it. And so you can switch between different behaviors if you have a suspension of particles, and you could go, for example, in this transitions from immotile to motile, for example. So that could be very interesting. And one final point is when we think about self-assembly in general, and in particular, hierarchical self-assembly. So we have a system that's assembling, and then it forms a larger structure, and then it forms a larger structure. And of course, in biology, this comes in a lot. And at all these levels of hierarchy, the systems are not really at equilibrium, and there are usually a lot of active processes that are happening because the system is alive. And so thinking about the context of hierarchical self-assembly, as we go up in the hierarchy, the size of the assembly, the assembled stuff is larger. And if this is happening in a fluid, then inertia will matter at some point as well. So these are just some things to motivate why this regime is interesting for active matter. And I should point out that really this has not been studied at all. This is all summarized in this Soft Matter article. It's a perspective that I wrote recently. It's very easy to read. So I will start with the first question, that is, what happens at the boundary? And in particular, what we're interested in, which I kind of alluded to, is can we design a swimmer whose behavior changes as we cross the boundary? So for example, if we have a swimmer or an object that doesn't swim on this side, and we gradually increase inertia, gradually increase the Reynolds number, at some point it should start swimming. How does this transition take place? What does that mean? And so on. So our model swimmer is a dumbbell, and it oscillates in time as shown here. So this is called a reciprocal swimmer because the first half of the cycle is exactly the same as the second half of the cycle in terms of spatially speaking, right? So in one case it's expanding, the other case it's compressing, but spatially they look identical. And so this is called the reciprocal swimmer. So this would not swim on this side of the boundary. Okay, Stokes flow, it doesn't swim. It would basically go back and forth. It would be like a scallop going back and forth. And this is I'm just going to show you a video just to get your intuition going into this. This was an experiment that we did a while back where we have this dumbbell swimmer, but we shake the container in order to get this to power itself. So it's a little different than a self-propelled swimmer, but this is what it looks like. And there's more details in this paper you can look at if you're interested, but here it is when it swims. So that was quite fun. I'm not going to talk about this a lot more than this. What I would like to say is that we are now looking at doing simulations of a dumbbell swimmer that is oscillating in time like this, and it is a reciprocal swimmer. And we're using what is called the Immerse boundary method with an adaptive mesh refinement. This is a software that's developed by Boyce Griffith and Chapel Hill, and there are a bunch of papers on this. I can talk more about this. All you need to know at this point is that this is a method where you resolve the fluid completely. So it's a computational fluid dynamics code, and it's quite fast, and it solves the full Navier-Stokes equations. And there are a lot of sort of nuanced things about getting these simulations to do what you want to set up these simulations. But this is the method, and I can tell you more about it. But I will go ahead and just show you some of our results. So here we have this dumbbell swimmer. It's going to oscillate back and forth. And what you're going to see here is that, so these swimmers are identical in terms of frequency and size and amplitude. And the only difference between these two swimmers is the fluid viscosity. Okay, so this is high viscosity. So this is a low Reynolds number. And this is a high Reynolds number. So this is more like water. This is more like honey. You can imagine it like this if you want something more intuitive. But what you see really quickly is that this swimmer is moving with the small sphere on the front, is moving down, and this one is moving up. And the flows that you're seeing here are basically the vorticity of the flow of the fluid around each swimmer. And you can also kind of tell that they're not identical, right? And I'll be talking more about this. But it was really interesting to see such a simple swimmer switch behavior, switch direction, just by changing the Reynolds number. So we did this. So we did these simulations for a large range of Reynolds numbers, and here's what we found. So this is the velocity of the swimmer as a function of the Reynolds number. And negative here means going down, positive means going up when it's aligned in this way. So what we found is the following. So at 0 Reynolds numbers, this is where Stokes flow is. So the assumption is that this is Reynolds number 0. There's no swimming, and this is good. We expected that because, as I said, this is a reciprocal swimmer. It means that if there is 0 inertia, it would just be going back and forth, okay? But as soon as there is some inertia in the system, what happens is that the swimmer moves with a small sphere on the front. So this is a small sphere leading regime, and it speeds up. So its speed is going up, and then it slows down, and then at some point around 17, it switches direction and it starts swimming with a large sphere on the front. Okay, 18, here it is. So even with this very simple model swimmer, we see this interesting behavior that was quite unexpected. We want to understand why. And in fact, we did this. So we repeated this. We did more simulations for a large range of amplitudes and radii, ratios, and distance between the particles. And what we found, this is a similar plot to the one that I was showing before, but a lot more curves. And what we saw was that we can actually collapse this transition if we use the right Reynolds number. So the Reynolds number is defined here. So now this is the amplitude of the small sphere. This is omega is the oscillation frequency. Little r is the radius of the small sphere, and here's the viscosity of the fluid. But what we see is that we can collapse this transition using this Reynolds number. So basically, these are the parameters that our system seems to depend on as far as the transition is concerned. So that means that is telling us that the transition is dominated by the small sphere, okay, because these are parameters of the small sphere. And while this is quite interesting, the next question I would like to ask is, what is the fluid doing? And I should remind you guys that the reason why we're doing a collapse and we're, sort of making this sort of conclusion from what we find is that the reason we are doing this is because there is no analytic solution to this system, either for a single swimmer. I'll show you what it is that we do know analytically, but this is something that has not been predicted or resolved, although there is some work, and we are also collaborating with applied mathematicians to figure this out, to understand this more. But looking at the fluid, what we did was to take the average fluid flow around a cycle of oscillation after steady state. And when we're in the small sphere leading regime, we found that the fluid flow looks like this. So there is the average fluid flow is pulling in the direction of swimming, okay? So the oscillation is in the vertical direction, and these arrows are the velocity field of the fluid, and these are streamlines. But what you see is that the flow is pulling in the direction of swimming and pushing out in the perpendicular direction, okay? And it's the same on the other side, so pushing out and then in. So this happens in the small sphere leading regime, and the swimmer is moving down overall. Now, at higher Reynolds numbers, here now we have the swimmer is moving with the large sphere at the front. What we find is that the average flow field is opposite in direction, essentially. So we see the flow is pulling in, perpendicular to swimming, and pushing out. There's this rotation here, these vortices. And there's a lot of complicated stuff that's happening between the spheres that I'm not showing here. It's not resolved enough. And that's also really quite interesting. But even the further flow over here, you can see that it has switched direction. And if you are familiar with Stokes swimmers, these look qualitatively very similar to pullers and pusher models. These are models for swimmers at 0 Reynolds number. So they're very different kinds of swimmers, but somehow qualitatively they seem, they're reminiscent. If you're not familiar with this, you don't need to worry about it. So 2 questions still remain. Okay, so one is, all right, like we see this flow field change, and it looks like it is related to the swimmer switching direction as well. But the question still remains, why does the flow change? And also exactly why does the swimmer switch direction? So these are still questions I would like to understand. So to understand this, we go back to breaking down the problem a little more to look at a single oscillating sphere. And this is a paper that was written back in '66, and it's an applied math paper where Riley is resolving analytically in 2 limits that I will show what happens when you have a sphere oscillating, a rigid sphere oscillating in a fluid at intermediate Reynolds numbers. And I'll take you through the way I understand this buildup of flow field. So this is what is called steady streaming. So we have a sphere that, so we're gonna go through the cycle of oscillation of a single sphere, okay? And basically what steady streaming is, this phenomenon is a non-zero time average, and I'll show you what I mean by this. So we have a sphere, a rigid sphere, oscillating in a fluid at intermediate Reynolds number. So when this, in the first half of the cycle, the sphere is moving up and it creates some vorticity behind it. In the second half of the cycle, the sphere is moving down and it creates these vortices behind it that are swirling in the opposite direction. If we were at 0 Reynolds numbers, so if there was no inertia or non-linearity, everything is time reversible, what would happen is that if we took an average over this cycle, the average fluid flow would be 0. We wouldn't see anything. However, here, because the system is nonlinear, these vortices persist. And in fact, in 3D, alpha and alpha prime is a vortex ring, is like a donut, and beta prime is a donut. Okay, so there's symmetry about the axis of oscillation, which is shown here as this vertical axis. But these vortices persist, and they also induce a secondary vorticity that is swirling in the opposite direction. Okay, so these vortices have to agree to be in the same direction over here, but the return flow is coming like this. So that means that this is counterclockwise, this is clockwise, right? So you have a vortex ring over on this side, alpha alpha prime, that's swirling in this direction, and then you have secondary vortex ring further out that is induced from the fluid there that is swirling in the opposite direction to the inner one. So the flow field for, so this is the average flow field around single sphere. So it is quite complicated. And these are stagnation points when the 2 vortex rings meet. What Riley basically showed is that this, so if we look at this inner vortex ring, so what I have in red here, if you were to, so this is, let's say, the general case. If you were to increase the viscosity, so you go from water to honey, this vortex would be larger in size, gets larger in size, and these outer vortices get further out and much weaker, okay? So what ends up happening in the limiting case that is the one that Riley studied mathematically, because that's the only thing you can do sort of with analytically, is that in the limit, still in the intermediate Reynolds range, what you have is you have this open inner vortex ring. It's no longer a vortex. But this is the same as the red one here, and you can see that it's moving in the same direction, right? So these are the pictures from Riley's paper for a single oscillating sphere, okay? So in one case, the oscillation is up and down and the fluid is being pulled up in. In the other case, the oscillation is up and down. There's this inner vortex, but further out, now the fluid flow is pushing out in the direction of oscillation. So we see this switch, and this is from Riley's paper. And now connecting it a little more to what we have been doing. So if you add another sphere, so now you create an asymmetry. Of course, we have this oscillation and there's the two spheres here. So the system is more complicated, but what I would like you to notice is that now we have these, we have this lower intermediate and higher intermediate Reynolds numbers. But if you look at a specific distance from the swimmer, in one case, the fluid is going up, and in the other case, the fluid is going down, right? So you have a switch in the fluid flows as well. So what we think is going on is that the fluid flow changes for the swimmer because it's related to this classical steady streaming results for a single oscillating sphere. Even though there's more things happening there, but maybe this is dominating. And the other question that I wanted to answer was, why does it switch behavior? And the answer is kind of that it has to, because the small sphere is like it's acting like a flagellum, while the large sphere creates a symmetry. And what I mean is that in both cases here, the small sphere is doing exactly the same thing. It's just going up and down. But in one case, at the lower Reynolds end, it's actually pulling fluid in and it's moving down. So it's like an appendage that's pulling fluid in and moving down. And in the other case, even though it's doing exactly the same thing, it's almost like it's pushing fluid away and moving up. So this was quite an interesting way to think about it. So the last thing that I would like to talk about in my remaining time is, okay, so we have these interesting sort of quite rich behavior from a single swimmer, traversing these intermediate Reynolds numbers. What happens when we have collective behavior? And so these are things that we have been working on more recently. And this was what I promised. I should say, as I said, like this is not as quite as neat as it looks. So no theory so far has predicted these transitions in the swim direction, and there's no theory for the shape of this curve. And so this is work in progress, and we'll have a paper soon for a single swimmer on this. But going back to, so looking at the collective behavior, so we have now started looking at pairwise interactions and multiple swimmers, and I'll just take you through what we have seen so far. So starting with pairs, what we found, we did a lot of, my student did thousands of simulations of different initial conditions for two swimmers. And what he found was that we basically see only these essentially 4 stable pairs that pop up. So one we call this in tandem V that I'll show you. So the swimmers align this V shape and then they move together, they swim together as one. We see these inline swimmers that again move together in one direction. We see these anti-parallel swimmers where they're head on and they sort of keep colliding and they sort of stay in place. And we also see these orbits, and I'll show you each one of those. So remember that now we are trying to understand, okay, so we know what the single swimmer does. We also have this transition in swimming direction. What happens when you have more than one swimmer? How does that change? And we might expect that when we have small sphere leading, so when the flow is pulling in the direction of swimming, pushing out in the perpendicular, we might expect that if we put another swimmer on this side, it will be pushed away. Like this would not be a stable configuration. Maybe more stable it would be, along this swimming axis. And it is in fact what we see. So this is for quite small Reynolds, so this is 0.5. So we see them forming this line and basically stay in place. Right, so it's not stable when it's on the side, but it aligns in this way. And if we increase the Reynolds number a little bit, we actually see this V shape, and then the swimmers swim together in this, we see this collective behavior essentially. And they are more sitting side by side, but even though the Reynolds number is still quite small. So here, if the single swimmer is moving with a large sphere on the front, you can see, you know, they do this stagger motion. It's a higher Reynolds number, and this one is a bit more complicated. We don't fully get this, so they sort of drift apart. I think it's more related to vortex shedding. This also looks sort of interesting. And we also see these orbits. So when the swimmers are moving with the small sphere in the front, they move counterclockwise. And then as the Reynolds number increases, they move clockwise. But the transitions, what I would like to say is that the transitions happen at different places, at different Reynolds numbers. So pairwise behavior is different from the individual. So this is the black line is the curve is the one that I was showing for single swimmer. This is where the transition happens for this swimmer. And these are the transitions for this V shape pair. Okay, so the transition from small sphere leading to large sphere leading happens at a different Reynolds number. For the orbit, it happens at a lower Reynolds number. So the behavior is quite complicated already as you go from 1 to 2 swimmers. And now if you add even more, you can really see the buildup of difference in behavior. So this is the system at a lower Reynolds, so Reynolds 5, this is 100. And already after a few cycles of oscillation, you see that here we're forming this sort of inline network type of structure, whereas here they do sit side by side, they form these triplets and sit side by side and swarm in a completely different way. Here, this almost looks like a stationary network. And this is from the paper that we are just submitting, which is this transition in the collective behavior from this network type of structure towards so swimmers aligning in this way along the axis of oscillation and of swimming to something that's more side by side, but still kind of looks like a network, to something where you have much more swarming type behavior. It's not particularly synchronized or organized, or it's not clear, at least yet, exactly if there is steady state here that we have access to. I have circled here these 3 junctions because these are at least one thing that we notice that is not predicted at all by pairwise interactions, right? So there are these 3 particle junctions that pop up. So I'm gonna just play you just a video of what these networks look like when you have more swimmers, or at the higher Reynolds number. You can see that the fluid is quite a mess, and I would just like to end with some conclusions and open questions. So one thing that I hope I have shown is that you can get pretty rich behavior from very simple reciprocal swimmers at these intermediate Reynolds numbers. And these steady streaming flows and hydrodynamic interactions in general, but in particular what I have been talking about, can induce very different structures and dynamics, different behaviors. A question that I think is very interesting is whether these steady streaming flows can be relevant for biological or artificial swimmers at intermediate Reynolds numbers. So can we utilize these flows? Are they actually utilized by biological swimmers? These are not things that have been studied a lot. Another question is what happens with other models. So I have only shown you one model, and there's already so much stuff to do. It's a very interesting question to ask. What are applications in materials? So this transition from going from immotile to motile, from non-swimming, so being in place like a pump, even the network that I was just showing before is not moving that much, going from that to something that's much more dynamic and switching from one to the other? Is there something, there are interesting applications in materials that we could think about? So with this, I would like to end. I will be very happy to answer any questions or clarification, anything you would like to ask. And I'm just going to leave these papers up and tell you that we're going to have a new paper soon on the pair interactions. And with this, I'm happy to take your questions. So thank you.
Right, so thanks, Daphne, for a very exciting seminar. And we have one little yellow hand up already. There's a question from Nunu.
Hi, Daphne. Thank you very much for your great talk and all these puzzling results at the end. So going back to your very simple reciprocal swimmer, what about the role of the mass of the different particles? So of the particles that make the swimmer?
Yeah, so this is a very good question. So in the model that we used, that I showed you most of the results for, these are the assumption here for these swimmers is that the density is the same as the fluid density. So this is like a, like a neutrally buoyant swimmer, where everything is kind of density matched. It's just, so in this case, we haven't really, I mean, the masses are different of the two swimmers because of the size. We haven't really played much in particular with the mass, but let me see if this is here, but the amplitude, where is it? Give me one sec. The amplitude of the swimmer, depends on the, so because of the way the setup is, the force on each sphere is the same. So it's equal and opposite. And so for the small sphere, what that means is that the amplitude ends up being bigger because the mass is smaller. So this is where mass comes in for the single swimmer. Now, what we haven't done is to have like two different densities of spheres, right? Or to vary the mass in a systematic way. But it would.
But my point is a bit simpler than that. I was thinking about the balance of forces on each particle, right? And so you have something that goes with the mass times in the case, so the largest acceleration that you get is something like A omega square, right? In the case of due to the oscillations, and you have a frictional force that scales with the size of the particles. So you have the same force acting on the particles, but the frictional forces are different on the different particles.
Yeah.
And as you evolve in terms of Reynolds number, this will change. So my point was, if at least you could have some insight about where the transition occurs just by looking to the balance between the forces.
So the forces become quite complicated because the drag force, what you're saying, right? So the friction, the drag force when you're in Stokes flow is just, is Stokes drag. And so exactly. It's very simple. When as you increase the Reynolds number, the drag force is not so simple. So it's not just, I mean, there is, of course, there is a force, and we can actually, solve for it numerically. So we can look at this force, but it's not, so there is a force balance, of course, but it's not super simple. Now, when it comes to mass in particular, maybe there is something you can say about mass, but I'm not sure. So the balance of forces is definitely, what matters. My point is that the forces are not so simple. Like this is
no, I get that, but I was just trying, at least in the low Reynolds number regime, to see if we did some Stokes friction and how it compares with the acceleration that you get, or the force that leads to the acceleration, internal forces on the dumbbell, if this could help at least understand why it starts to go down and why eventually it starts to go up.
Right. So this is the issue. So when you're in Stokes flow, I think this is a better plot. When you're in Stokes flow, the assumption there is that, I mean, you have your Stokes drag and that's it. Okay. Now, as soon as you are not in Stokes flow, you have more complicated things happening. What you can do, and what we have sort of done, and this is kind of what the applied math paper did for a single sphere, is like you can try to, well, yeah, in a different context because there was no swimming there, but you can look at what happens, like almost if you perturb around your Reynolds zero, would you get a prediction that the swimmer would be moving down? Or, this is a harder thing to predict. This is harder to predict because the Reynolds number is higher, but over here, could you predict that the swimmer will move down? Yeah, as it turns out, we can sort of do that, but it is not, yeah, it is actually quite complicated, because to resolve this, you do need higher order terms. Is not something that you can get so easily from doing very simple theory, I guess, which is why we're working with applied mathematicians who are doing, like, I don't know, very complicated fluid dynamics analytically to figure this out. So I think, this is what I would say.
Again, because my point is, think about this scheme. You'll see that the force, the internal force is the same on both, but the frictional force is lower on the bottom one. So I understand this somehow tells me that why they go down at the beginning, right? Because when it moves down, it's in the limit that you have a very big object at the top, is the one in the bottom is the one that is moving. Mm-hmm. And so
but why would it move down?
Because every time it goes up, then you are trying to compress a liquid in between two obstacles, and when it's moving down, it's just moving down. And this is the one that moves easier, then it's trying to go down. But I don't know, I'm just thinking a lot.
Yeah, you're right. Yeah, this is the compression. This is a good way to think about it. I don't know that, this is a good way to think about it, but yeah, I mean, that's all I can say. I mean, I'm not sure. I don't know that. I don't think that if you write this down, you will get this result, but it is,
it's okay.
Like a good way to think about it in a way. Yeah, it is a bit more complicated in the end. Yeah.
Okay, thank you.
Right, thanks for that. Any more questions? Oh, Margarida.
Just a minute. Okay, I've unmuted myself. Thank you, Daphne, for a nice seminar. I have a question about the two swimmers.
Mm-hmm.
Of course, when you have the two swimmers, you showed that the short range flow is different, and it depends. It gives you transitions at different values of your number, your whatever number, whatever you called it. But did you try for the two swimmers to see if there is something for different? I didn't understand if for the two swimmers you tried different parameters, or if this is just a set of two different swimmers. When you say that this is different from the one swimmer, are you changing the same type of parameters? Or
yes, yeah, we're just changing the viscosity of the fluid, let's say. So we're changing exactly the same thing. The only difference is that instead of one, we have two, and we put them close by, and then we see what happens.
Okay,
so in the one case, when you have one swimmer, what we see, so this is the black curve here, we see that there's a transition from going down to going up that happens at some, Reynolds number. And then if we have two, they sit side by side, and then the curve looks quite different, and they move down in this case, like with the small sphere on the front. And then at some point, this happens. So there's a transition and they start moving up. But this transition happens at a different Reynolds number. And also, you know, there's all of this stuff happening here, which I didn't talk about much.
Mm-hmm. Yes, so my question is, yes, of course, this is different if you just change the Reynolds numbers and keep everything else constant. But are these type of transitions, when you have two swimmers, universal in some sense? So if you changed other parameters, could you try to see if this type of transition, which is different from the single swimmer, for the double or for the pair of swimmers, still scales in some way?
Yeah, it's a very good question. Like if you look back at, so for the single swimmer, we varied a lot of stuff and, you know, saw this transition. And we haven't done that for the pairs, actually. And in fact, I think what we are getting closer to now with the analytical work that we're doing is to understand really what, so this is not necessarily exactly what will end up, like I said, this is a collapse that we think is what's going on, but this is not quite the whole truth of why, of what controls this transition. So the system is actually pretty complicated. And there are, you know, different terms that dominate in the different Reynolds numbers. And so this is what we have seen analytically. And now for the pair, we haven't varied a lot of things yet. But what I would like to do is, once we have a better grasp of the analytical solution, then at least having, you know, knowing, you know, having a function of knowing what this system depends on, we can vary parameters in a wiser way.
Okay.
And it would, of course, the analytical thing is for single swimmer, but at least would be better informed for what to vary for the pairs as well. But I think your question is, yeah, it's a very interesting one. And I should also say, which I didn't talk about much, and these are all the nuances of this particular system, which is that there are different dimensionless ratios that one can define. So this is one in particular that I defined, and I defined it because of this reason, because of this collapse. But actually, you know, it doesn't have to be, there are many different dimensionless ratios that actually one, strictly speaking, should be keeping track of, and they're not independent of one another. So you can't be, you know, in Stokes, you know, you can't have like really, really small Reynolds number of one definition and really large in the same system and really large of another definition, right? So they're all close by, but keeping track of all of them and seeing how, you know, how you can change your behavior based on changing a parameter. So for example, I guess maybe what you're saying is that, you know, given this curves that I have here, if you have this Reynolds number, you can cross this boundary by changing your frequency, right? Or changing your amplitude. And this is kind of true, but it's not completely true. Like you couldn't go, and this is where the analytical results are very helpful.
And one other question, which is related, of course, you talked about the single swimmer as a pusher or a puller. So if you have many of them and look just at the long range flow, you would expect to restore some kind of maybe complicated but more universal behavior.
Yeah, so what we're seeing is, so if we look at the average fluid flows for the pairs, there's also, so we are thinking of them in this sort of pusher puller kind of context, just because it's easier. But again, here it's also a little more complicated. So if you see these are, so you're increasing the Reynolds number, moving to the right. So so the average fluid flow here is this is sort of stationary. So the average fluid flow is like pushing out here, pulling in. So this is a bit like a puller, but a stationary puller. And now you have this V shape moving down, and this is pulling over here, but pushing out here. So it's not really a puller in this case. And then it kind of becomes a polar as you increase the Reynolds number more, but it's also a bit messier because it's moving around. And so it does become more complicated. There are some things that in this paper that we're submitting now, we haven't gone into great depth, but we are looking, well, we kind of have, but we're not describing it in the paper just because it's like there's too much. But what we're looking at is also like the average fluid flow for a single swimmer and for the pair are not, you know, always obvious. So in some cases, it's sort of, they're the same. It's just like adding another particle. And in some cases, it's something more complicated that happens that, you know, seems to be the stable thing, you know, the ground state of the system, but we don't exactly know why necessarily. Yeah.
But can you classify that when you get more or less the same by adding another swimmer and when you have something completely different?
Yeah, what do you mean classify?
No,
sorry, what do you mean?
Change, I mean, you're just changing Reynolds numbers, right? So it's difficult for me to understand what is really changing, except that the local flow or the short distance flow is different.
Yeah, so I mean, as you increase the Reynolds number, all that's changing is the viscosity of the fluid, but that has an enormous effect on your system. So the fluid is, so it's both, you know, short range and long range. I'm showing quite a close up here, but yeah, so the viscosity of the fluid, the result in terms of the physics is that there's, you know, there are more and more non-linearities that add up. So this is what this inertial term does. But the result of it is not obvious at all. And this is what I'm sort of showing here. And what we are trying to do is to build from the single swimmer behavior, to build an understanding for the pairs and then for the collective behavior. So one of the things that, you know, one might sort of predict in a way, in a hand wavy way, is that when you have an average fluid flow that is pushing out here and pulling in here, on average, if you put another swimmer side by side at these Reynolds, it wouldn't want to sit there. It wouldn't be stable. It would be pushed out. Of course, the other swimmer also has this fluid flow around it. So it's not obvious what happens when you put two. And this is in fact what we see, but there is some understanding, some intuition that you can get from the single swimmer, but it is definitely work in progress. It's not, yeah. So there are some things that kind of make sense. So you have more inline swimmers here. Here, you have more swimmers that are in line, which kind of makes sense, but you also have these triple junctions that you can't predict. As you increase the Reynolds, you get more side by side, which one could have predicted from the single swimmer case. But again, the fact that, you know, hydrodynamic interactions are many body interactions.
Yeah.
So it's not, yeah, it's not necessarily obvious, or it's all things to be figured out. Yeah.
Thank you.
Right, okay, very good. Any more questions? Looks like there are no more questions. So if there are no more questions, then I think it's time to wrap up. Thanks again, Daphne, for a very interesting talk. Many thought-provoking issues. Thanks to everybody for coming, and we'll be back on Thursday of next week. So see you then.
Thank you. Thanks so much.
Recording stops now.
Bye-bye.